In our final video in the pH buffers topic, we're going to talk about arguably the most important application of pH buffers, that being the regulation of the pH of human blood. Now, regardless of whether you're a biology expert or not, most people understand that blood exists to transport oxygen from the lungs to the cells of the human body, and on the reverse trip, transporting CO2 produced as a waste product from cells back to the lungs so that we can exhale it and get it out of the body. Now, blood's ability to do this is regulated by and influenced by three different equilibrium systems. The first of which being the equilibrium between the gaseous form of CO2 that is produced from reactions in the body and the ability for that CO2 to dissolve within water. Therefore, the equilibrium exists between the gas form and the aqueous form of CO2. Now, once dissolved in water, most CO2 does not stay in the CO2 form. It undergoes a synthesis reaction with water, forming carbonic acid. And likewise, carbonic acid does not simply sit in water unreactively. It undergoes an acid ionization reaction in water, forming hydrogen carbonate and hydronium as a weak acid. Now, to understand the way that these equilibria shift, because it all starts with the production of CO2 gas, we need to go to the source. Now, most carbon dioxide in the body comes from this single reaction here, which biology students will obviously recognize as cellular respiration. This is the chemical reaction that is used to produce the energy that our body needs in order to survive, with CO2 as a waste product. This means that the more energy our body requires, the more cellular respiration we're going to do, meaning the more CO2 is produced. Now, if we follow this across all three of these equilibria, we can see a problem on the large scale that develops. So as our body does more cellular respiration to produce more energy, the concentration of the gas form of CO2 is going to increase, meaning that in this equilibrium, the equilibrium will shift to producing more of the aqueous form, and therefore we're going to have more CO2 dissolved within our blood. Now, if we have more CO2 dissolved within our blood, that's going to push the synthesis reaction equilibrium in favor of the creation of more carbonic acid. So as the amount of CO2 in our body increases, we would expect the concentration of carbonic acid to increase as well. Now, if this is true, if the concentration of carbonic acid is going to increase, we would expect in the acid ionization equilibrium that it will shift the equilibrium to the products, and therefore the concentration of hydronium is also going to increase as well. And because hydronium indicates what the pH of our blood is going to be, we would expect that this pH will drop and our blood will become more acidic as we produce more energy. Now, biologists will immediately recognize this is a problem because acid is very, very dangerous when it comes to the effect on the body's proteins. Since this isn't a biology course, we won't get into the mechanism of this, but the short of it is that high concentrations of acid naturally damages protein structure in a process that we call denaturation, the verb for which is the verb to denature. And basically what acid does is it causes the complex structures of our protein to unfold so that all of the proteins lose their functions. Now, when we consider that all muscle in the body is made of protein, and most of the biological catalysts, the enzymes that speed up our body's chemical reactions, are all made of protein, this means that any large-scale pH decrease is going to be very, very dangerous for our body's survival, which is why we need a buffer in order to stabilize our blood's pH. So, the solution to this, get it, it's a pun because the acid is dissolved in water anyway, it's best that we forget about that. The solution to this problem is to actually use the carbonic acid that we make as a buffer itself, because if we have carbonic acid, we can also make a large supply of its conjugate base simply by ionizing this acid. Because whenever we have carbonic acid and hydrogen carbonate, we have just made a buffer solution, provided that these two exist in a similar concentration. However, because carbonic acid is a diprotic acid, we can also make a second buffer solution if we take carbonic acid and ionize it to form the carbonate ion like so. So, which buffer system does your blood actually use? Well, that depends on what the pH of the buffer solution is, and that is relatively easy to figure out if we assume that the concentration of each acid and its conjugate base is equal, meaning that we are at the transition point, meaning that we can calculate the pH by simply taking the negative log of the Ka value of each of our acids. So let's go and check the Ka of carbonic acid and of hydrogen carbonate. So carbonic acid, which we find right here, has a Ka value of 4.3 times 10 to the negative 7, while hydrogen carbonate has a Ka value of 5.6 times 10 to the negative 11. So we can simply check the negative log of each of these numbers. So if we check the Ka of carbonic acid, we can see that the pH of a buffer solution at the transition point is approximately equal to 6.4. Now, 6.4 is still a little bit acidic, but it's pretty close to 7. So right now, the carbonic acid carbonate or bicarbonate buffer is looking pretty good. So let's check the second Ka value, the Ka value of hydrogen carbonate, which is the negative log of 5.6 times 10 to the negative 11. And if you put this into your calculator, this comes to approximately 10.3. So this pH is definitely too basic because it's quite a bit farther away from 7, meaning that our concentration of hydroxide is too high in this case, and too much base can cause just the same amount of problems as too much acid can. So we can see by checking the pH of each of these buffer solutions that the carbonic acid and hydrogen carbonate buffer is a lot more appropriate to select as the buffer solution in human blood in order to keep the proteins within the human blood stable. This concludes our series on not just pH buffers, but also the applications of acid-base equilibria. So to make sure that your understanding is clear, make sure that you try all of the practice problems below, as well as all of the other practice problems concerning acid-base equilibrium.